Welcome to Bryophytes. This week we're just going to focus on bryophytes. The reading is chapter 16, all of it, but focus on what I talk about here today so you can focus your reading, because a lot of the things in that chapter we're not going to be paying attention to. Specifically, I wanted to talk about bryophytes before we move on, because bryophytes are still in the Archaeoplastida, right? It's still one of the evolutionary lineages that resulted from the original endosymbiotic event. So that's why we're talking about it now. But where we are right now is we're kind of in this basal section, right? We're talking about the changes that happened when protists developed other traits. And some of these traits allowed some of their descendants to live on land, but not all of them. So your learning objective for today is to understand how mosses or bryophytes are similar to algae. How are they similar to plants? And what are the major groups of bryophytes alive today? There's only 3 of them, so I'd like you to know a little bit about them, and I'll talk about those at the end. Okay, so pretty much it's the same theme that we've been talking about throughout the class. How many ways are there to be a protist, right? These organisms have figured out how to live everywhere on Earth except for land. We haven't talked about how they've developed a way to live on land. Sure, there are protists that live in the soil pores and things like that, but as of yet, we haven't talked about any adaptations that facilitated a permanent transition to land. Now, mosses, the way I think about them, they're just a different kind of lifestyle for photosynthetic protists. That's it. They're nothing that different. And when you start looking at them and taking apart their bodies, you'll see they're pretty simple organisms. And we've seen organisms like this already in the class. So I talked, I don't know if it was last week or the week before, about how colonial algae is kind of a solution to multicellularity without having to commit to multicellularity. And that makes sense if you're an aquatic or marine organism. Doesn't really happen. It doesn't work on land, right? Here is a slide of tissue from a bryophyte or moss. And here's one from a colonial protist. I mean, they look almost the same, but the advantages of living together as a colony, you need water, you need moisture, you need transport among cells. You can't do that on land. So this wasn't an option. So multicellularity has advantages, but you can't do it in a colony on land. And so that's how I think of bryophytes. They're a way to live on land for a really simple organism, a really simple photosynthetic organism. Really, the only difference between bryophytes and protists, they're just more specialized. That's it. They have very few but very specific specializations for life on land to prevent drying out and to facilitate reproduction. And I'm going to talk about them. Bryophytes, mosses, I mean, where do you find them? You will find them, by streams like this photo. You find them in wetlands. You find them in wet areas that have standing water at least sometimes. You don't find them in arid climates. And that's because they're not yet plants. They're not fully adapted to desiccation and to drought. They don't have vascular tissue. They don't have roots. They don't have real leaves, how we define them. They don't have stomates. Now, stomates that's probably one of the biggest ones. They have very, very poor exchange over their control over the gas exchange, right? So when they can't close their stomates, and stop losing moisture from their bodies when it dries out, they just keep them open. And that's why you don't find them anywhere where it's really dry, because they would just lose all their water. They can't control it. So they're not yet plants because they're not truly adapted to dry conditions. Now, although bryophytes, the ones alive today, aren't transitioned, they don't represent the true ancestors of land plants. They are descended from a similar ancestor a long time ago. They represent a transitional lifestyle between algae and land plants. Now, this is an old one, right? We've got charophytes as being along the evolutionary line, but from our paper last week, we know that that's not quite right, at least not the way the molecular data tells us. But the rest of it kind of makes sense, right? We have bryophytes, give rise to simple plants, the lycophytes, the manilophytes, the gymnosperms, and the angiosperms. We're not going to talk about the plants. That's for a different class. We are just going to talk about how mosses are similar to plants. So I said they're different from plants because they're not adapted to life on land, but they've got some really strong similarities. First of all, the way their chloroplasts are made. The thylakoids are in grana, they're stacked. So the chloroplasts have stacked and very specific function, very different from bacteria and lower other algae. So stacked chloroplast, grana. And this is what I was waiting for this week to talk about. Where should I put my picture? Okay. The way that they divide the cell cytokinesis is different, right? So here's a picture, and they talk about this in the text, but this isn't from your text because I like this picture better. So look at it this way. On the left side of this picture is an alga, and on the right side is a vascular plant. And it shows how they have very different ways of cell division. In the algae, the microtubules align to pull apart the daughter nuclei, but the cell invaginates. So there's cleavage furrowing, and that's how the plant initiates cell division. Whereas in vascular plants, this is like a more recently evolved trait, there's no cleavage furrow, there's no invagination. Instead, the microtubules initiate phragmoplast, which is cell wall formation between the daughter nuclei. So when a cell divides, they actually lay down cell wall material, and that's how cell division progresses. So algae, when we think of it, algae are more like this. Vascular plants are all like this, but some algae are intermediate, and mosses are more like plants. So one of the traits people use to figure out which algae are most likely ancestors of plants or how do they divide their cells. And this is one of the traits that charophytes share with mosses and vascular plants. Okay, so bryophytes and plants both have phragmoplast cell division. They both have an apical meristem, growth that's initiated by a set of undifferentiated cells at the top of the plant. And you can see how this one might be adaptive for life on land, right? If you're growing up, right? There's a directional growth. The sun is up, away from the ground, which is less important if you're growing in a three-dimensional aquatic environment. Another similarity between bryophytes and plants is they've got motile male gametes. So male gametes are the only cell that's motile in these organisms, and their flagella are similarly oriented out of the sides of the body. So I just wanted to show you this picture because male gametes in plants and lower plants are crazy. They're just the most wild looking cells. They're so beautiful. But there's two flagella on these cells. Now, perhaps the most important similarity between bryophytes and land plants, and a reason that bryophytes were able to be successful in land, is because they have oogamous reproduction. And we've talked about this a bit. Oogamy means that the female sex cell is much larger and is retained in the gametophyte, and the male sex cell is small and motile and is released. This is a picture of just a generic reproduction in bryophytes. Their reproduction is very, very simple. I'll go over it in a little while. But the key is they have oogamy, true oogamy, and they all do. And this is true for plants also. But maybe, so oogamy is important for transition to life on land, and I'll talk about that in a minute. But maybe the biggest development in the way these organisms work is the transition to matritrophy. This is matriarch, maternal. This is mothering. They mother their embryos, and this hasn't been seen before. So the embryos are taken care of by the gametophyte generation. So the gametophyte generation actively feeds and protects and nourishes the babies. And this is the same as embryophytes. This is the same as land plants. And this is huge because it allows the sporophyte generation, the diploid generation, to have a head start and to be developmentally advanced enough that it can survive on its own if it needs to, where it's a bit drier. It doesn't always have to survive on its own in the bryophyte. Sometimes it's retained on the gametophyte, and I'll show you. But this was a huge innovation. Okay, so the reproduction of mosses is pretty simple. There's sporic meiosis, so both generations are multicellular. They're anisomorphic, so they look different. But what's really, really important is that the gametangia, the way they produce gametes, they've got elaborate structures. And for the first time, we see archegonia and antheridia. And these are structures that that persist in the plants, true plants. But archegonia now is the name we give to female reproductive organs in the mosses, and antheridia are the male reproductive organs. And they're on the gametophyte generation, but they're multicellular, so they can produce lots of sex cells. So here's a good picture of the antheridium, right? And this is the male sex organ. And you can see it's full of many, 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 many cells that is going to produce a lot of sperm cells. So in some of the other protists, the gametes are single cells, right? They just produce single cells and that's it. They produce hundreds and if not thousands because they have specialized gametangia. So these specializations just make it more likely they're going to survive on land. And here's the archegonia, and the archegonia are just these little outgrowths. They're multicellular, but they're not huge. And they've got a single egg cell in the bottom. So it's got this neck canal cells, the ventner, it protects it. And you've got one single egg cell. So fertilization happens by transfer of sperm cells to the venter. And we'll talk about that in a minute. But the point is, these organisms, bryophytes, have specialized gametangia like plants, which allow for more gametes to be produced. It allows for the gametes to be protected while they're maturing. And really, really importantly, it allows a place for fertilization to occur. And this is why I said oogamy is such an important development for transition to life on land, because fertilization doesn't happen in the open environment. It happens in the safety of the gametophyte, and it happens in the archegonium. So the egg doesn't ever leave the archegonium. It's never out there in the environment ready to dry out or get eaten. It's safe within the gametophyte generation. And this was a really big development. Okay, so, sorry. Okay, so these are all the ways that bryophytes were similar to plants, but how are they different than protists? And in your text, they talk about charophytes, so I'm going to talk about charophytes because charophytes do share a lot of traits with bryophytes. How are they different? Because we've seen some of these traits in the algae, right? So how are bryophytes different than algae? How are they not algae? Well, let's talk about the retention of the zygote in the female gametophyte, right? I talked about the retention of the egg, which we've seen before in protists. But what we haven't seen before is, right, I already talked about that. Sorry. What happens is the neck cells of the canal, they disintegrate, they create a fluid-filled tract for the sperm to swim down to fertilize the egg. So it all happens, internal fertilization the whole way. I mean, some people wonder, why do land plants, especially like a desert plant, why does that need motile sperm? Well, take 210. We talk about that a lot in Biology 210. But in a lot of these organisms, the sperm isn't delivered directly to the egg. It still has to swim through the female gametangia to get to the egg. And that's what happens in bryophytes. Okay, so post-fertilization, the gametophyte, the zygote develops inside the gametophyte. It isn't released into the environment. The calyptra is the enlarged ventner. I talked about the ventner as a little group of cells basal to the egg. It grows along with the zygote and it provides protection for the young sporophyte as it grows. right? And if you've ever looked closely at a moss and you've seen these sporophytes sticking up and they've got these little yellow caps, that's just the relict of the venter. That was tissue that was inside the archegonium to protect it. And it protects it as it develops and as the sporophyte grows out of the gametophyte. Okay, so what's different here now is the embryo is turning into the sporophyte entirely within the gametophyte. And this is a cross-section through an archegonium, but the embryo is highly developed, and you can actually see the whole baby plant. The embryo is developing inside of the gametophyte. You can see the foot, the seta, which is the stalk. You can see the capsule, that's going to be the sporangium. But you can also see the placenta, and that brings us to the matriotrophy, right? The fact that these plants actually nurture their embryos. So the placenta, just like in animals, it's a tissue that's at the interface between the embryo and, in this case, the gametophyte, which isn't the case for animals, forget that. But okay, let's have a better look. So placenta is made up of sporophyte and gametophyte cells, right? So you've got the gametophyte, the archegonium, and you've got the baby sporophyte, the embryo developing inside the archegonium. And what happens is extensive wall ingrowths in the placenta, in this tissue, and it just represents the tissue joining the mother and the fetus. Here, let's, I have a better picture. They talk about this quite a bit in your text, right? Okay, so if this is the gametophyte, this is the placenta, you can see all the ingrowth, which facilitates transfer. It's apoplastic, so basically the nutrients are just kind of leaked out of the gametophyte and into and absorbed by the sporophyte. They don't have really specialized structures, not like they do in animals, but they have these transfer cells in the placenta that are really invaginated. And the upshot is the sporophyte gets nutrients to grow before it's photosynthetic, so it can leave the gametophyte generation or not leave the gametophyte generation and be quite independent before it has to photosynthesize by itself. So matriotrophy is just the mother supplying, or the gametophyte in this case, supplying nutrition to the developing embryo. And it was a big innovation. All right, so if you look, if this is the archegonium, you see the structure and you can see the neck cells and you can see the embryo developing, right? It's got probably several hundred cells at this age. The placenta is just this little section right here at the interface of those two, right? So this is a bryophyte, this is a marchantia, and you can just see the wall ingrowths intermingling and facilitating nutrient transfer. Embryophytes. So embryophytes is a term we give to bryophytes and vascular plants because they nurture these little embryos in the gametophytes. They have multicellular matriotrophic embryos. And what this matriotrophy does allows for this establishment of this independent sporophyte earlier, but it allows us to get bigger and produce more myospores, right? Because meiosis happens in the sporophyte generation, and if bigger it is, and the more advanced it is, the more established it is, the more myospores it can produce to produce more gametophyte generations. So here is this is an idea. Like if you look at all photosynthetic organisms, starting from algae all the way to flowering plants, there is a difference in the anisomorphy of the generations, right? Early on, the gametophyte generation is dominant, and most of what we see in the environment is a gametophyte, and it's got very tiny or very reduced sporophytes. But as evolution progressed, the sporophyte got bigger and bigger, the gametophyte got smaller and smaller. Right now, at bryophytes, they're about equal. The sporophyte and the gametophyte, I mean, the gametophyte's a bit bigger, but not by a lot, compared to algae. In some algae, the gametophyte is huge and the sporophyte's a single cell, or compared to flowering plants where the sporophyte is billions of cells and the gametophyte is 7 cells. Right now, we're even Steven. They're very, very similar. Okay, but like I said earlier, it's sporic meiosis, so you've got multicellularity in both the gametophyte and the sporophyte. And even though they're similar in size, what you see in the environment is the gametophyte. Because the sporophyte, sometimes it isn't even chlorophyllite. Sometimes it's just reproductive structure that pops out, and that's all you see. When you're looking at moss, chances are you're looking at the gametophyte because it's dominant. Now, what kind of mosses are alive today? Well, there's 3 lineages of bryophytes: the liverworts, the true mosses, and the hornworts. And I just want to give you some sense of what these groups are like, so you can go out in the world knowing something about them. Liverworts are so cool. They just look otherworldly. They look kind of oily and chubby. They're really beautiful. Mosses, I think what most people think of when they think of mosses are Bryophyta, the true mosses. But then there's also hornworts, which are a little bit different and a little bit scragglier. They kind of look like lichens. Let's start with Marchantiophyta. This is the liverwort phylum. And like I said, they're these weird little oily little creatures that you sometimes, the only time I ever see them, except for like in a rainforest, is in potted plants. Sometimes I'll get a plant from the greenhouse and I'll have some liverworts growing in it. They're so beautiful. There's many species, about 5,000 species, but they're all really simple. Now I said it's mostly gametophyte. What we're looking at is a gametophyte generation. The sporophyte is so reduced that it's never actually released from the archegonium. This is an archegonium in the gametophyte full of sporophytes, and these sporophytes basically are just little capsules of spores. That's all they are. So they're very reduced, and they're entirely embedded in the gametophyte, and they're only released when the gametophyte breaks down. Spores are only liberated when the gametophyte thallus dies. So when the body of it dies, that's when they're released. So this is an example. Even though I said they're even Steven, there's variation among the bryophytes. Here, the gametophyte is very, very dominant. So when you're looking at a liverwort like these guys, you're looking at the gametophyte. They also have asexual reproduction, really common. Fragmentation, the thallus will just get fragmented and start growing again. Or else they've got these little gemma cups that are just asexual, little modular little organisms that can germinate and grow into a whole new thallus. Okay. So like I said, the sporophyte's very reduced in this group, and either it's embedded within the gametophyte entirely, or it could be these little tiny sporophyte, right? These little tiny umbrella-like creatures sticking out of the gametophyte, grows out of the archegonium, but never truly leaves the gametophyte generation. I mean, in some cases, they're really reduced, right? You've got this whole leafy liverwort, and then you've just got this tiny little sporangium peeking out, and that's all you get. That's the only part of the diploid generation. Right? So if you look at the drawing of the moss in the middle, everything except for that little stick, the sporangia, everything else is gametophyte. Sporophytes only a little bit on top. Okay, that's Marchantiophyta. Bryophyta, the true mosses, there's many, many. There's like 10,000 of them. We're only going to talk about 2 of them, Sphagnidae and Bryidae. There's 3 more we're not going to cover. They're not covered by your text. I just want you to know a little bit about them. Sphagnidae is obviously the group that peat moss belongs to. Sphagnum, you've probably heard of them. They're found all over the world in boggy areas, and they've actually been really culturally important. Well, they're ecologically important in peatlands in terms of the global carbon cycle. It stores a huge amount of carbon, right? Peatlands are really, really deep, and they form slowly over millennia by compression of all the plants. And they produce a form of carbon because it's compressed over millennia that has been used traditionally as a fuel source. Here's a wetland, beautiful. Right here, pioneers harvesting peat, and looks like the 1800s, right? They just dig it up. These little bricks were like bricks of fuel, and they would burn them for heat because they're really carbon dense. So it's a huge source of carbon. The Sphagnidae have very distinctive sporophytes. They've got these spherical capsules on top of the gametophyte generation, which is the green leafy part. And they've got a spectacular spore discharge. And let me see if this works. Okay. As the sporangium dries out, it creates pressure up to 5 bars. That's the amount of pressure in a truck driver. It can move 16 meters per second, and move up to 12 centimeters. So it is pretty spectacular. Okay. Now, sphagnum is been culturally important because it's really absorptive, and that's due to the unusual leaf anatomy. The leaves are only 1 cell thick, and there's only 2 cell types. I told you, like, bryophytes are not very specialized. So they got hyaline cells, which are these big empty ones in the middle. They're dead when at maturity, and then you got the small photosynthetic cells at the margin. Now, the effect is these big empty cells store a huge amount of water. So sphagnum is very, very absorptive. It can hold 20 times its dry weight in water because of these hyaline cells. And for many, many, many millennia, they were used as diapers. Here's a picture of a Dene woman using moss diapers as recently as 1947 in Manitoba. Where's another picture? A Cree woman harvesting moss in 1927. I mean, it's been used by humans for many, many years, but maybe not as extensively as it is now. And harvesting peat moss is a huge global industry for horticulture. And it's not pretty. I mean, you know, every time you buy some kind of potting mix, it's from sphagnum, it's from bogs, it's from peat. And Canada is huge in peat harvest in Canada, but the problem is it only grows 1 millimeter a year. A peat bog, which is, you know, meters and meters thick, took tens and thousands and even more years to be created, and we're harvesting it. It's not exactly a renewable resource given the timeline of our harvest cycle and given the population of the planet. So we are destroying our global peatlands. And the problem is not just we're going to lose this really special ecosystem, but these peatlands store 100 times more carbon than even tropical forests. They're a massive carbon reservoir. So in terms of global climate change, harvesting peat is really, really problematic. Not only that, but the peatlands that we have, with global temperature increase, they're going to start respiring, decomposing, and all the stored carbon from millennia is going to be liberated into the atmosphere. So again, climate change has all these unanticipated feedbacks, right? Feedback loops where we're going to start losing even or putting even more carbon into the atmosphere than we predicted because of these changes. And one more thing about sphagnum, bog people, right? You guys might have heard about bog people. They're always, you know, especially in the United Kingdom where they have a lot of bogs and they've had people living there for a lot of people living there for a long time. They find people who were either put in bogs or died accidentally in bogs and were covered by peat. It preserves them so perfectly because of the phenolics in the cell walls, and they decay very, very slowly. So really intact people with hair and clothes are dug up from peat bogs. You can learn a lot about old cultures this way. Yeah, it's kind of gross, but very interesting. Okay, true moss, class Bryophytes. These are the true mosses. This is what you're thinking of when you probably think of moss. They're called true mosses because they've got branching filamentous protonemata, and we haven't talked what about a protonoma is yet, or nemata in plural. But protonemata are the first stages of gametophyte development in the bryophyte. So when the myospores germinate and are on their way to becoming the leafy gametophyte, what does that look like? And it's called a protonema or protonemata. And in the true Bryophyte, in this class, they are branched. And yeah. And it looks very much like a little plant, right? It looks like there's root structures, even though they're not roots. It looks like the leaves, even though they're not leaves. It looks like a protoplant. Now, I said earlier that they don't have vascular tissue, and they don't have true vascular tissue, but they have these cells that are specialized, that are kind of function like vascular tissue, even though they're not actually vascular tissue. These are called hydroids and leptoids. Hydroids act like xylem, leptoids act like phloem. So hydroids conduct water, leptoids conduct sugars. They're not true vascular tissue because they don't have the same chemical composition. They're not lignified, they don't have secondary tissue. So we can't officially call them vascular tissue, and yet they are. Okay, so I said they don't have stomates. They can't control water loss from their leaves, and so it makes them very vulnerable to drying out. They have not real guard cells, but they have specialized cells to regulate gas exchange and water loss, and not very responsive to the environment. In this case, they're only one cell, whereas in true plants, there are two cells for each stomate. So they're not true stomata, and they don't have very good control over gas exchange, but they have a little bit. They have more than algae because of these cells. Okay, hornworts, Anthracerophyta. Hornworts, there's not very many of them. There's only about 300, and they're called such because they have this horn-like sporophyte. So this thing sticking up, that's the entire sporophyte generation. The little fuzzy leaves on the bottom, that's the gametophyte generation. So here's a picture of it from your text. The sporangium at maturity just splits open to release the spores, but it's still maintained in the leafy thallus-like gametophyte. These guys are very different from the other bryophytes we've looked at so far. They have true stomates. So this group alone has two cells that open and shut depending on the water status of the plant. So when the water status is too low, they shut to preserve water in the plant. And when there's tons of water, they open up so they can facilitate photosynthesis. Like I said, they don't have a lot of control over these stomates. They're only responding to water availability, not to light, not to humidity, not to all these other things that true plants, their stomates are really quite complex. These ones are really simple. So they work, but they're not great. The other thing this group has is they have dichotomous branching versus rosette branching, right? And this is similar to earliest land plants, this dichotomous branching. And they have meristematic region of the sporophyte. So this is very similar to the sporophytes we showed you before, right? It's in the archegonium, you've got the placenta, you've got the foot, you've got the seta, here's the sporophyte developing, but this one maintains meristematic tissue at the base of the seta. It doesn't fully differentiate like it does in other mosses. So this means the sporophyte can keep growing as long as conditions are good. The other mosses, the sporophytes have a very fixed growth period, and no matter how good the conditions are, the sporophyte can't keep growing. This means they can respond to the environment. This is more similar to true plants that have a lot of meristematic activity, and they can respond really quickly to changes in their environment. That's it. That's all I have for today. I hope you enjoyed the introduction to the bryophytes, and I will talk to you in a few days.